for question 2, I believe last week I gave you time to assemble in class and you've taken some photos to put it in. Okay? But just like recap where we left off, you have to put your mouth down if you want to eat the chocolate this morning. Okay, you were asked to join several nucleotides together, right? And if you refer to your notes, this is how the nucleotides will join together. Okay, if I do a quick zoom in, we have a look at the structure of a nucleotide. Okay, just a quick recap on the structure of a nucleotide. Number 1, contains a sugar. Number 2, contains a base. Number 3, contains a phosphate group. Your task last week was to join the nucleotides together. We find that the nucleotides join together at the phosphate group and where carbon number 3 is, the hydroxyl group. Okay? If you have not taken note of this, if you want to go into your notes to make a quick point over here, it's very important for you to know how the nucleotides join together. Okay? So you either go into your notes or go into your CA to make a note that this is where they will join together via condensation reaction. You're expected to know this. Okay? Okay, so on the right hand side is what it looks like if you join many nucleotides together. What I expect you to know is that they join at carbon 3 and a phosphate group that is at carbon 5. Okay, today, this morning, we're going to do a quick activity. Okay, I'd like everyone to grab, not everyone, maybe in groups of 2 or 3, grab one of these bags. Okay, I'm going to pass it around. You can keep it with you. You work as one group, huh? You work as one group. You work as one group. They work as one group. Alright, this morning, here we have a plastic sheet. I'd like you to take out the large plastic sheets inside. Okay, take out the large plastic sheets inside. Okay? And you'll find the large plastic sheets are labeled A, T, G, and C. Okay, could you take that out? A, T, G, and C. Okay, before we begin today's activity, just a very quick note on the structure of DNA. Here, I'm a bit lazy this morning, so I printed this out and made magnets. I'm going to show you the rough structure of a DNA first this morning so that we understand what we're doing. Okay, first and foremost, here I have a strand, a DNA strand. It's just a single DNA strand, right? Here are all my nucleotides. You can also see very roughly the sugar over here, the base, and the phosphate group. Notice the only thing that changes is the base. I can change the base, but everything else is the same. The sugar, the phosphate group, the sugar, the phosphate group, and the bases. For those that have done 4B, you have learned that DNA is not just a single strand. Right? A real DNA molecule consists of 2 single strands interacting with each other. So here another strand. I'm going to put it on the bottom. Okay. Often, scientists describe DNA as kind of like a ladder, right? Look like a ladder, yeah. It kind of looks like a ladder with two backbones, and with the bases as each rung of the ladder. Right, so it kind of looks like a ladder over here. What do you notice? What patterns do you notice when you look at this particular DNA ladder? Any patterns you see, the way I have constructed this?
The G always goes with the C, and the A always goes with the
I notice that the G always goes with the C, A always goes with the T. So I want you all to make a mental note. Write this somewhere in your notes. And this is very important. You need to know that G always pairs with C, and A always pairs with T. Okay, we call this pairing complementary base pairing. When we say base, we are referring to A, T, G, or C. Complementary means you complement each other. So complementary base pairing. How do I use this in a sentence? I would say that the bases of the DNA complementary base pair to each other. Or inside these two DNA strands, we observe complementary base pairing. Okay? So this is how I can use it in a sentence. I say again, between these two DNA strands, we observe complementary base pairing. Or I can say, there is complementary base pairing between the two DNA strands. Okay? So that's how I can use it in a sentence. Okay, next structure of DNA. I'd like you to pay attention to this. Right, notice that for every strand of DNA, single strand of DNA, there will always be one end that is still free, that has a free phosphate group. Right? And then the other end will have a free hydroxyl group. Where's this hydroxyl group? It's at carbon number 3. Okay? So there will always be one end that's free hydroxyl, one end there's always a free phosphate group. Just because we haven't joined any nucleotides at both ends. Yeah? So there will always be a free end, right? Okay, so we refer to the end with a free OH group as a 3' end. The apostrophe on top is read as prime. Okay, 3' end. Some students will ask, why do we call it the 3' end? Because the hydroxyl group is bonded to carbon number 3. If you don't believe, you look over here, carbon number 3, 1, 2, 3. That's where the hydroxyl group is bonded. That's why we call it the 3' end. On the opposite side, we call the free phosphate end as the 5' end. Okay? Why 5? Because that phosphate group is bonded to carbon 5. Right? So, as a biologist, one thing that's important for you is to know how to count the carbons. Which I found is not our strong suit for carbohydrates. I will make sure I go through in depth how to count the carbons for your corrections video. Okay, so there's a 5' end and a 3' end. Yes?
The G don't have E, man.
The G don't have
E.
E.
The P.
Or the P. Okay. So Hao-Yang is holding up the big plastic things I've given you, right? Okay. What I provided you does not contain the sugar. Does not contain the phosphate group. It is purely the base. Okay. What do I mean by that? You see a apostrophe with an H over there? Yeah, I see a apostrophe with an H. That is where the sugar is supposed to connect to. But I don't want you to focus on too many things. Okay, it's just the bases. Okay, so now bottom. This end is called the what end? Okay, the bottom DNA strand. 3' or 5'? 3', huh? 3' end. And the other side? 5' end. Do you notice that the strands run opposite to each other, the direction? One goes from 5 to 3, one goes from 3 to 5. The second characteristic that I'd like to introduce to you, the word is called anti-parallel. DNA is often described as anti-parallel. What does it mean to be anti-parallel? One strand goes in the 5' to 3' direction. The other strand in the 3' to 5' direction. That's what it means by anti-parallel. Okay, two characteristics I brought across so far. Number one, there's complementary base pairing. Number two, it's anti-parallel. Maybe number three, the most obvious one that I kind of just gloss over is double-stranded. Okay, DNA is double-stranded. Double-strand means there are two strands. The last thing I would like to introduce, which will lead us into the next activity, is what in the world is holding our two DNA strands together? Right? It's literally like putting two chopsticks together. They can just fall apart. Right? But as you know, when you receive a fresh pair of chopsticks, they're all stuck together, right? So how do the DNA strands hold themselves to each other? Okay? Actually, they hold themselves together by a particular bond you've already heard of before. It's called hydrogen bonding. So between the bases, there's hydrogen bonding. So I'd like to add on to what we've learned just now. The bases, complementary base pairing through hydrogen bonding. I'd like to show you now the structure of the DNA. We've been building carbohydrates so far, right? But today I brought one that I built in my staff room. This looks super long to build. Okay, this is the structure of a DNA. You see the black colors? Okay, black represents what atom? Carbon. White represents? Hydrogen. Yeah. And red represents? Oxygen. Okay. Purple is a new color. See the purple color over here? The purple represents, let me guess, phosphate. Okay, so have a look at the DNA model. I'm going to try balance in one hand. Okay. So you look at the DNA model, notice that the sugar is on the outside. Right? The sugar forms one chain like a backbone. Right? So if you look at this black part over here, black, purple, and then it goes round. Right? It's like sugar, phosphate. Actually, we call this a sugar-phosphate backbone. Okay? There are two sugar-phosphate backbones. Can you see? Here is one sugar-phosphate backbone. This one backbone. Another backbone over here. If I go down this way, you see? Two backbones. So where is the backbone of the whiteboard? The backbone is simply this segment over here. Okay, minus the bases, that's the backbone. Right? If you ever counter this word, don't be afraid. Sometimes they call it a sugar phosphate backbone. Okay? At the core of the DNA, you'll find all the bases. Oh. Okay? You see at the middle, you see a lot of blue. What element is blue? Represented by the blue atoms. Nitrogen, okay? So that's where all the bases are. You find the bases interacting with each other at the core of the DNA. Somehow, something is holding the bases together. The two backbones aren't just unzipping and falling apart, right? Somehow they are interacting with each other. That's hydrogen bonding. So, this morning we're going to try to figure out how the bases hydrogen bond with each other. If you look at the next activity of our class activity, let's go to this part of our class activity over here. And we'll give the front part a quick read. It says that adenine and thymine can form two hydrogen bonds with each other. Okay, what does that mean? So between A and T, there is two hydrogen bonds. Between guanine and cytosine, you expect to find three hydrogen bonds. The task for today morning, what am I saying? The task for today, the morning of today, is to find out how the two bases interact with each other by hydrogen bonding. In your pairs or in your groups of three, I would like you now to find the bases AT and GC. Using your knowledge of how hydrogen bonding should work, you are now tasked to arrange the AT and GC until you can form 3 hydrogen bonds or 2 hydrogen bonds. You are allowed to flip the bases in any way you want. You can reorientate them in any way you want until you can arrive at 3 or 2. That is the puzzle we'll try to solve this morning. Okay, how young got a question?
That one is what?
Sorry?
That one is what?
1.8. Yeah? Okay? I would like to give you this morning around 7 minutes. Okay, not 7 minutes, 10 minutes. 10 minutes to figure out this puzzle. How do I form 3 or 2 hydrogen bonds between the groups? Okay, your time starts now. Yeah. Orientation. Okay, you look at the screen now. I'm just going to show you a sneak peek. Okay, so you can interact something like that. Okay, so your task is you try out first. Okay, once you think you figure out how they interact, you can look to your textbook to see how it actually interacts. Okay, in the last 3 minutes, okay, I'd like you now to try the configuration in the textbook. Okay, I'd like you to try the configuration in the textbook. Arrange it until you can get this 2 hydrogen bond and 3 hydrogen bond, so you can have a look yourself. Okay, you will need to flip things around and rearrange it in order to achieve this. Once you've got the arrangement, I'd like you to snap a photo. Once you manage to mimic the arrangement. Okay, snap a photo. Okay, once you snap a photo, I'd like you to draw in your hydrogen bonds. Okay, you have until 8:30 to set your photos, include into your CA. Then by yourself, try to join the hydrogen bonds too. Okay, practice where hydrogen bonding can take place. Okay, until 8:30 to complete this task. A minute more. Say again. Oh, that helps link up the how to see the doctor that unique strength is I haven't seen told you what the name is. Unique strength is their bonds to each other, right? And across the strands it's like bang. Alright, your time is almost up. Okay, can I draw your attention to the whiteboard? kinds of bonds we're looking at in the DNA. The first kind of bond is this. When I want to join a new nucleotide, okay, can I get everyone looking at the whiteboard? Okay, when you join a new nucleotide, right, phosphate group, carbon number 5 to carbon number 3 at hydroxyl group, if I were to join a new nucleotide, it's a condensation reaction. This bond over here that links them together is called a phosphodiester bond. Okay, I expect you to know this bond. Just like how between two sugars, you can say it's glycosidic. Between two amino acids, it's peptide bond. Your triglyceride, there's an ester linkage. You need to know the bond in DNA. It's a phosphodiester bond. Yes, hi.
I thought you say it's hydrogen bond. How come we come that one?
Yeah, so that's why I'm bringing this up, because I noticed there's some confusion. Okay, so phosphodiester bond links nucleotides together. Okay. Next kind of bond involved, all the hydrogen bonding. Okay, hydrogen bonding allows the bases to interact with each other. Okay, so there are two kinds of bonds here. One is hydrogen bond is between the two DNA strands, the bases. Phosphodiester bond is within the strand, linking the nucleotides together. Yes, Henry?
But you know, like the G and the C, there's 3 hydrogen bonds.
Yes.
Then how come the C and the G got 1 hydrogen bond?
Oh, because I just haven't added on.
Oh.
Okay, yeah. Thanks, Henry. Yeah, for those that missed it again, phosphodiester bond links the nucleotides together. So, in order for me to join this nucleotide, I need to form a phosphodiester bond. Hydrogen bond links the bases together between two strands. Okay, so there are two different bonds. Often students confuse between the two. Sometimes they think that what I'm using to join these two together is hydrogen bond. Then they think that these dotted lines are phosphodiester bond. Okay, it is the other way around. So, if you're wondering where you can find this in your notes, okay, you scroll to this particular page over here. Okay, notice that they've labeled the phosphodiester linkage. Phosphodiester bond or linkage, same thing. Okay, bond or linkage is the same thing. If I were to ask you to circle a phosphodiester bond, you want to circle the entire phosphate group. I say again, if I ask you to circle a phosphodiester bond, you would circle the entire group over here. The P with the 4 O's, that's a phosphodiester bond. Okay? Okay. Okay, I'm going to summarize the structure of DNA right now. Okay, ready? Let's summarize the structure of DNA. Number 1, DNA. Maybe that should be number 1. Here, DNA is double-stranded. That's the first characteristic. Number 2, the two strands are anti-parallel to each other. That means one strand goes from 5 to 3, the other 3 to 5. Third characteristic, the two strands interact with each other through what we call complementary base pairing. And it's linked together via hydrogen bonding. The final characteristic is that every nucleotide is joined to the next nucleotide via phosphodiester bonds. Okay? So these are the characteristics of a DNA. I will bring this up over and over again so that we get more and more used to it. Quite a new vocabulary that we encountered today. Okay, so I give you some time now. Take down your notes, touch up your notes as I set up the whiteboard for the next segment of the lesson. Okay, I will not be doing the rest of 4A. The rest of 4A is extra. Okay, it is for your information. If you want to stretch yourself a bit, you want to know a little bit more about the chapter, you can do it. But it's not within our syllabus. Okay. I'll be moving instead to B, 4B. So once you're done, you can go to 4B. We're just going to compare DNA and RNA. If your group is done, can I get you to pack everything back into the plastic folder? Okay, so an open call. The competition for this year is up. Okay, those who want to join the competition, you can join in pairs. Your task will be this. I'm going to disassemble this today. And if you want to, you can work in pairs and sign up in our class chat. The winners will get a free meal from me. Okay, the task is simple. Okay, no, it's not simple. The task goes like that. You have to assemble this in 1 hour. Okay, it's possible. I assembled this in around 50 minutes by myself. Okay, you work in pairs if you want to. Okay, you get a free meal from me. So these are clues. This is a structure of DNA. If you want to construct it, you want to find that there are 2 sugar phosphate backbones. Okay, you also want to find that the strands are anti-parallel. See over here, I have one free phosphate group, the P. If I were to go down the opposite end, you'll find that there's a free, oh my god, okay, there's a free OH group at the other end. At the top over here, there's also a free OH group. If I were to follow this all the way down, you will find that there's a free phosphate group at the end. See, it's anti-parallel, right? So that's one clue. Next clue, you know that between GC and AT, there are different number of hydrogen bonds. You look over here, I have actually 2 bonds over here. So the A and T and the G and C interact with different number of bonds. That's another clue for you. Okay? So if you're interested to sign up, I will be leaving this at the atrium in a box. The challenge goes like that. You will find a partner and then you will set up your phone and then you will time-lapse yourself building it. It is possible to build. Okay? If everyone cannot hit under 1 hour, and it's also fine. I will still treat the fastest team to a meal. Okay? So this is a competition within our class. Okay, fastest team to a meal. But if you want to, or if you can, try to hit under 1 hour. That is a real challenge. Okay? Okay, I'll leave this in class for you to analyze. So that after you analyze, you feel that you're confident, you want to sign up, you can sign up. Okay? Great. To end off today's lesson, we're going to compare DNA and RNA. Okay, we're going to compare DNA and RNA. Your homework over the weekend was to watch the videos so that you can compare DNA and RNA. There are a few points of comparisons that I'll be making on whiteboard today. Yeah, let's compare DNA and RNA. take down this roof. Yes?
When we're doing like the challenge, can we like refer to the source like
Can refer to your textbook. Yeah, you can refer to your textbook if you want to. You use any material you want. Some students are, okay, no, I shall not tell you how they hold their parts. Okay, you are asked as your homework to compare DNA and RNA. Actually, the fastest way to compare, right? If you went to YouTube a little bit more, you will find that actually people have done comparisons before, DNA versus RNA. Okay, expect to be able to that you may be asked how to compare DNA and RNA. Today, I spent a bulk of the lesson talking to you about the structure of DNA, right? But you must be able to also compare RNA to whatever we talked about this morning. Okay? So let's do a quick comparison between DNA and RNA. Okay, your homework was to compare. Here they are going to list a few. As they list out the comparisons, I would like you to take down as much as possible. If you cannot, then you can go home and also search for this particular video. Yeah?
You're writing clear and concise, no matter where you are. Compose a cover letter.
Thanks for stopping by. Let's talk about the differences between DNA and RNA. So DNA is usually double-stranded. It has two separate strands that are connected, which look like this. While RNA is single-stranded most of the time, which look like this.
Okay, point number one. Yeah? DNA is often double-stranded. That's what you see on the whiteboard. RNA, I usually find them as single strands. Okay, so what would be useful for you is to draw a table. Okay, to construct your notes. You can do DNA, RNA. Then normally, how I do a comparison table is that over here I have characteristics. So the first point could be about a single / double strand. Right? So that's how I would make my notes. Okay, to compare point by point. Your carbohydrates one, they ask you to compare between the glycogen and all the stuff. Also possible to draw tables. So nice and sticky. Look like it's quite colorful. Last time when I was in school, the time teacher is talking, right, I will take my eraser, then I'll erase. Then I'll gather all the eraser shavings, then I'll make glue tag out of it. You know, you can make glue tag out of eraser shavings. I'll make a lot of it. Sometimes I'll buy one whole eraser just to erase away so I can make a glue tag. Okay, so first point up. Second point.
DNA is much larger than RNA, with usually millions of nucleotides. And RNA is, of course, smaller, with hundreds to thousands of nucleotides.
Okay, next is in terms of length. What I show you on the whiteboard is it's not very fair. Doesn't quite do DNA justice. DNA is made up of lots and lots and lots of nucleotides, millions of them. RNA is really, really short compared to that. Even this model I have over here is a really short segment of the DNA. DNA is extremely long. Next.
Sometimes tens of thousands. DNA has a deoxyribose sugar, that's why it's called deoxyribonucleic acid. Its sugar structure looks like this. The DNA, of course, has the phosphate group and the nitrogenous base. And And this carbon here is missing the oxygen. As you'll see with our RNA, which has a ribose sugar, looking like this, again with the phosphate group and the nitrogenous base. And this carbon here, the number 2 carbon, has an oxygen attached to it. This carbon
Okay. Third difference, the nucleotides of RNA. If you look at the sugar, at carbon number 2, it has a hydroxyl group. You don't find that in DNA. Okay. Specifically, if you want to describe it, it's at carbon number 2. Carbon number 2, you find an extra hydroxyl group for RNA. Okay, it's at carbon number 2. Okay, third difference. 2 more difference.
here, the number 2 carbon has an oxygen attached to it. The location within the cell for DNA is primarily located in the nucleus, with some DNA being in mitochondria as well.
That's not how you spell nucleus, yeah?
For RNA, while some is in the nucleus, most of your RNA is in the cytosol and in the ribosomes throughout.
Cytosol, or cytosol, cytoplasm, cytoplasm. You often find DNA in the nucleus. RNA, you often find it in the cytoplasm. Last difference. By the way, you all know how to embed a video into your OneNote? Yeah? So, actually this is something you could do also. Okay, you can embed this video into OneNote and then you don't have to technically copy everything. Number 5.
For your nitrogenous bases, DNA has 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. And RNA also has 4 bases: adenine, guanine, cytosine, and uracil. So RNA does not have uracil, or I'm sorry, does not have thymine, but has uracil. So RNA does not have uracil, or I'm sorry, does not have.
Okay. Yeah, RNA does not have thymine. I believe that's not how you spell thymine either. Okay, so remove the A. Okay, so RNA does not have thymine. Instead, it replaces thymine with uracil. So every time you see a U, what RNA is trying to represent is trying to represent a thymine, but it doesn't have thymine. Okay, if you're done, can I get you look up? Okay, so in summary, What's the difference? Number one, RNA tends to be single-stranded. This is what I'm trying to represent on the whiteboard. Number two, you often find RNA in the cytoplasm. Number three, if I were to look at the sugar of RNA, okay, if I just look at the sugar part over here, at carbon number 2, you'll find one more OH attached to it. Okay, next point, you often do not find thymine inside RNA. Instead, all the thymines are replaced with uracil. Okay, so that's one big clue that it's RNA and not DNA. That's the last point. Location, single-stranded, the size, thank you. Okay, last one is the size. RNA is often very short. Okay, it's not very long. Okay, so 5 differences if you're asked to compare DNA and RNA. Right. Can I get you to touch up your notes for the topic of nucleic acids? Okay, you can go to your notes, and if you scroll down to the segment of RNA, you could include these points of comparisons between DNA and RNA. Okay, so scroll a little bit more. They've also provided you some comparisons. This is where you could include your comparison table. That's all for today. So after you compare, you can put it inside your notes. So I do not need you to submit 4B, right? But what I needed you to do was to run through the comparison process yourself. Okay? Do you manage to get some of these points of comparison as you were doing it yourself? We made it it's a little bit more challenging for you because you watched 2 separate videos. Okay? Other than that, 4A. Okay, 4A is the one I would like you to submit. Okay, could you submit 4A? 4A, you only need to complete up to 4C over here. Okay, you only need to complete up to 4C for submission. Yes?
How do you change the one to light mode?
Oh, okay. You change your entire iPad. I think your iPad is in dark mode now. Is it?
Oh, yeah.
Yeah. Okay, so for users who are using iPad in dark mode, when you export the image of your worksheet, I don't know why it will turn gray. Why? Okay. I supposed to have lessons with you tomorrow, right? You're going to have HBL. Okay? So you go and have a happy HBL, go and have your passion pursuit, whatever it is.